Back in the 1950s, scientists had a big question. When cells copy DNA, do they keep the original, mix it up, or split it in half? Now, imagine this. You unzip a jacket down the middle and hand each half to a tailor. Instead of making two brand new jackets from scratch, each tailor uses the half you gave them to completely build something new. Half old, half new. Enter, ladies and gentlemen, the semi-conservative model. The method your body uses billions of times a day to keep life going, one strand at a time. Now, let us first have a recap. What is DNA? DNA is made up of nucleotides, each with a phosphate group, a 𝐑𝐞𝐳 sugar, and a nitrogenous base. These bases pair specifically with A and E, G with C, forming a double helix. We use a special mnemonics. Apple is a tree, car is the garage. Okay, we have two questions for you. Now, the first one is, what are the three pairs of a nucleotide? Okay, you got it right. Now, let us proceed to the second question. Why is base pairing important for copying DNA? Exactly. Now, what is DNA replication? DNA replication is how a cell makes an exact copy of its DNA. It happens during the S phase of the cell cycle and ensures genetic information is passed to new cells. Okay, let us answer another question. What could happen if a mistake is made during replication? That is correct. It could either be mutation, a disease, or a malfunction. Let us now proceed to the semi-conservative replication model. In DNA replication, there are three proposed models: conservative, dispersive, as well as the semi-conservative. A conservative mechanism of replication proposes that all DNA is used as a template only and is not incorporated into a double helix. Thus, the new cell has one completely new double helix and one completely old double helix. The dispersive model, on the other hand, posits a final product in which each double helix of DNA is a mixture of fragments of old and new DNA. The semi-conservative model is the one that is correct. Each new DNA molecule has one old and one new strand. Watson and Crick proposed it, and Meselson-Stahl confirmed it through their experiment. Let us answer another question. Why is keeping one old strand helpful? That is correct, because it serves as a template and ensures accuracy. Let us now proceed with the mechanisms of DNA replication. In the process of DNA replication, the DNA makes multiple copies of itself. It is a biological polymerization, which proceeds in the sequence of initiation, elongation, and termination. Furthermore, it is an enzyme-catalyzed reaction. DNA polymerase is the main enzyme in the replication process. DNA replication in prokaryotes has three main phases: initiation, elongation, as well as termination. Although it is similar in eukaryotes, the initiation process is more complex in eukaryotes than prokaryotes. In eukaryotes, there are multiple origins of replication present, but as for this discussion, we'll solely focus on prokaryotes. First, we have the initiation. It starts at a specific site on the DNA called the origin of replication. Helicase breaks down the hydrogen bonds between the base pairs, unzipping the DNA, forming a replication fork. As the strands separate, topoisomerase prevents the DNA ahead from getting too tightly coiled. Single-strand binding proteins hold the strand apart to prevent them from re-annealing. Primase lays down a short RNA primer. This is crucial because DNA polymerase can start from scratch. Now, what would happen if helicase didn't work? Okay, you got it right. Without the replication fork, no replication would happen and it can't begin. Now, let us proceed to the elongation phase. This is the phase where actual copying happens. DNA polymerase III adds a new nucleotide in a 5' to 3' direction, reading the template 3' to 5'. On the leading strand, this is straightforward and continuous. The lagging strand, however, is built in short fragments called Okazaki fragments because it's synthesized in the opposite direction. A sliding clamp keeps DNA polymerase attached to the DNA. DNA polymerase I later replaces the RNA primers with DNA. Ligase seals the gaps between the Okazaki fragments, forming a continuous strand. Okay? Now, to test your attention, let us answer another question. Why is it harder to replicate the lagging strand? Okay, you got it right. Because it's made in pieces, not in one smooth line. Okay, let us now proceed to termination. Eventually, the replication forks meet and the process ends. Thus, proteins stop helicase from going further. Topoisomerase 4 helps separate the newly formed circular bacterial DNA molecules. Proofreading during DNA replication involves scanning and correcting errors at the 3' termini of nascent of the DNA strand. This is performed by the 3' to 5' exonuclease activity of DNA polymerase, which removes the mismatched nucleotides so the correct base can be added by its 5' to 3' polymerase activity. In bacteria, DNA polymerase 1, 2, and 3 all possess this proofreading ability. When a mismatch is detected, the polymerase reverses by one base, excises the incorrect nucleotide, and inserts the correct one before continuing replication. Let us now proceed to the Meselson-Stahl experiment. This experiment proved the semi-conservative model of replication, where each DNA molecule has one O strand and one U strand. Let us know the procedures behind this experiment. Meselson-Stahl grew Escherichia coli bacteria in a medium containing heavy nitrogen-15. The DNA became dense. They then moved the bacteria into a medium with light nitrogen-14 and allowed the DNA to replicate. After each generation, they extracted DNA and spun it in a centrifuge to separate DNA based on weight. And the results show that in the first generation, DNA formed one intermediate bond, which disproved the conservative model, which would show one heavy and one light bond. In the second generation, DNA showed two bands, one light, one intermediate, which disproved the dispersive model. This supported the semi-conservative model, where each new DNA helix has one old and one new strand. Alright, let us answer our last question. If replication were conservative, what would the result be? Now that is an exact answer. One heavy band, which is the original DNA, and one light band, which is the new DNA. To summarize what we have discussed today, let us have a wrap-up. DNA replication ensures genetic continuity. Now, the semi-conservative model produces one old and one new strand. Its key enzymes are the helicase, primase, DNA polymerase, and ligase. The Meselson-Stahl experiment is the one that proved the semi-conservative model. Now, here is a reflection question for you. Which part did you find the most interesting in our discussion for today? The enzymes, the experiment, or the copying process? How many did you get from that quiz? We are hoping that you got a perfect score. Now that is it from all of us here at G-nius Review Center. And again, today we discussed the semi-conservative model and we hope that you learned something new.